Mom's a math teacher.
Okay, you guys, so let's go over your homework. Number one, so they give you a generic, they give you a generic equation, chemical reaction. A is converted to B and C, and the stipulation here is that it tells you that they're giving, you have a continuous supply of A, and so therefore you don't run out of A. So that remains constant over time. That's important in answering the question. So number 1A asks you that you compare two side-by-side experiments of this reaction. And experiment 1, you use X amount of the enzyme that catalyzes this reaction. And experiment 2, you use 2X amount of the same enzyme. Well, how does that affect the reaction? How does that affect the reaction? What do you think, Shane?
The reaction rate would be doubled.
Yeah, the reaction rate would be doubled. So it increases or doubles because you have double the amount of enzyme and you have this continuous supply of A. All right, absolutely. B, you compare two side-by-side experiments. You use both equal amounts of the enzyme, but now in one experiment you allow the products to accumulate over time, and one you remove the products from the system as they are produced. And so in this particular experiment, how do each one of these affect the reaction? Allow them to accumulate over time, and you remove the products from the system. What do you think, Nathan?
In experiment 3, the reaction rate would have a gradual decrease in slope because it has to spend more time looking for reactants over products. And experiment 4 would have a linear rate because you didn't have to look for reactants to the products.
That's right. All right, absolutely. So the graph here for the rate, this is the rate of reaction over time. So that first one, eventually, as Nathan said, the slope would decrease until eventually, will decrease because decrease there because you have the accumulation of the products, and so just like in the toothpick activity, you could pick out broken toothpicks. So the more products there are, that will slow the reaction rate. And then if you remove them, we talked about this actually on Friday, that every single time you pick, you're going to pick out a substrate or a toothpick that's not broken, and so therefore it'll be a linear, increasing steadily at its maximum rate. All right, C. In the next two experiments, you use equal amounts of the enzyme, but this time you change the temperature. One is at 20 degrees Celsius and one is at 25 degrees Celsius. So in this particular case, what could happen to the reaction rate? Devin, what do you think?
The experiment as opposed to like the optimal temperature would react faster.
That's exactly right. So we don't know. They didn't give you any information about the optimal temperature. And so you can't say that it'll occur faster at 25 degrees versus 20 without knowing. So as Devin said, the one that's closest to the optimal temperature will be or have the faster reaction rate. Could it be that both of those temperatures are outside of the realm of temperature range? Absolutely, because when you have a temperature range in which an enzyme can work, you might have, if this is the temperature and this is the rate of reaction, so you might have, so let's say this is your optimal temperature, it gets too hot and it drops down, all right, and here's, it gets colder. If the temperature is right here, then even right here versus right here, it might be that both of those, 20 and 25, might be outside of the optimal range for that particular enzyme. And so it could be that neither one of them work, but we just don't know because we don't know what the optimal temperature is. So that goes, kind of flows right in with letter D. In letter D, you're looking at pH. So you're still using this equal amounts of the enzyme, but now we're changing the environment and doing pH. And for the same reason for the temperature, we change this to pH. It depends upon what the enzyme's optimal pH is. And so if its optimal pH is around 5, the rate of reaction would be faster at pH of 6 than it would be at pH of 8, but we don't know what the optimal temperature or optimal pH is. All right, so then number 2 talks about how enzymes can be inhibited or regulated. So A asks you to compare competitive and non-competitive inhibition and how they differ. So, Chad, what do you think? Non-competitive versus competitive?
Competitive reaction lower, non-competitive like doesn't.
Well, non-competitive, it actually also does slow down the reaction. What does it do? Why is it called non-competitive? Do you remember where it binds to the enzyme? All right, right, and so that might, when you think about it, the active site, not the inhibitor not competing for the active site, you might think that it doesn't slow the reaction down. But it actually does, and why does it slow the reaction down? Maddie?
Because I think it's called the Allosteric Site or something, and it binds to that site, and it makes the other shape completely unusable.
That's right, so in non-competitive, and this is your enzyme, we can have a site different than, if this is the active site, we can have a different site on the enzyme that is not the active site, that's called the allosteric site. And so in competitive inhibition, if this is your substrate here, and the substrate can fit into the active site. In competitive inhibition, the inhibitor, it can fit into the active site. So this maybe could be the competitive inhibitor. So this can also fit into the active site. So what happens is if you have both of these in the cell, they're in competition to get to that site. If the substrate gets to the site first, then the reaction can occur. The substrate will be converted to product, whereas if the competitive inhibitor gets there first, then the substrate's out of luck and the reaction isn't going to happen. So that is competitive inhibition. But when we look at non-competitive inhibition, this inhibitor binds to that other site. So the substrate is still the same shape and can fit into the active site. The non-competitive inhibitor will bind to this allosteric site. So let's say it has a shape like this here. So this is non-competitive inhibitor. If this gets there first, what it does, as Maddie says, is when this binds there, you're right that the active site is still free and clear, but what it does to the active site is changes the active site's shape. So when that non-competitive inhibitor binds, the active site changes shape, which means now that even though the active site, there's nothing there, the substrate still can't bind to it. And so this is non-competitive inhibition, and it also decreases your rate of reaction. So both of them decrease your rate of reaction as a result of that. And so, so B, we haven't really got to allosteric enzymes, but allosteric enzymes are enzymes that have an allosteric site like this. That's a site separate from the active site. And what it does is regulate the enzyme, because what happens is if you have an inhibitor that binds to this, it changes the shape of the enzyme, which means that that reaction, converting that substrate to product, isn't going to occur. And so we'll come back to this after. We're going to do a little bit in our notes today about allosteric enzymes, but it's basically a way for your cells to make reactions happen or not. And so, if in your cell you need to convert the reaction or the substrate to product, then your cell will make it so the enzyme is in the active shape and you won't have an inhibitor, and vice versa if we don't want the reaction to occur. All right, and so, like I said, we'll come back to that. All right, and lastly, number 4, you have this generic M pathway. And this is a pathway where we're taking A and eventually converting it to molecule D. And so it's A gets converted to B, which gets converted to C, which gets converted to D. And each one of these reactions are catalyzed by a different enzyme. And so, our overall reaction is A gets converted to D. But remember we said, like in cellular respiration, there's many small steps in between, each catalyzed by a different reaction. So they use an analogy of this, of making shoes. So A is the leather, and the leather is cut into parts, so that's reaction 1, and so we have our parts that are needed for the shoe. Then it says the parts are sewn together, so that's reaction 2 here, catalyzed by a different enzyme. And then, so they're sewn together, and then making C, and then they're packaged for shipping, which is now we get D. So now they're telling you that, so that's your factory, that shoe sales are decreasing. So shoe sales are going down. So you're part of this factory, so and increasing in backpack sales. And so what the manager decides to do is to slow down the shoe making because they're not selling as much shoes and increasing the backpack. So this is the shoe making. And so where would the manager want to stop this reaction or this process from happening? Probably at the first step, right? Otherwise, it would be a waste, so we want to stop here because it would be a waste of resources. They're using the leather and energy by the workers to do any part here if we don't want to get the shoes to the store to make the sale. So we want to stop at step number 1. So then relating this to a cell. So if this is a reaction, if this is a reaction in the cell is doing this process to get molecule D, whatever molecule D is. So molecule D is important for some use in the cell. So this molecule is going to be used for whatever reason. And so then it's telling you, though, that, so the cell is making D, and this is supposed to be used, and that's why it's expending energy to do that. Then it says if a cell is in excess of the chemical product D. So what does that mean? So let's say the cell, for some reason now, doesn't need molecule D. So it's been making D, all right, and D is now starting to build up because you're not using D. So you got a whole bunch of Ds in our cells. And so what happens is that then the cell wants to stop this reaction. So what occurs? This molecule D, we want to stop it for the same reason as that we stopped the factory at step number 1. We want to stop the reaction at step number 1. So the cell isn't expending resources and energy making all these intermediates if it doesn't need D. So this molecule D actually stops its own production. And how do we regulate this? So what does D actually do? So you've got all this molecule D in the cell. What does it actually do? Well, to regulate reactions, what makes reactions go faster in the first place? Enzymes, right? So what happens is molecule D actually regulates this first enzyme. And in this case here, do we want this reaction to occur? No. So what do you think molecule D does to the enzyme? It inhibits it, absolutely. And so we're going to see that the actual parts of the reactions can actually inhibit and control their enzymes, which can control the rate of the reaction. And that's what we're going to look at here today. All right? And so letter C, when it asks you what type of enzyme does this, we're going to look at this today here. These are called allosteric enzymes. All right. So we're going to talk about this more in depth now. All right? So we're going to wrap up chapter 8 for today. So we'll have it, you guys, some people have been asking me about tests and so on. I'm combining chapter 8 and chapter 9. We probably won't start chapter 9 until tomorrow. And with chapter 9, we have a lab that we're going to do with cellular respiration. You're going to be kind of designing your own lab to see what kinds of things affect the rate of cellular respiration in certain organisms and so on. So we're gonna look at that. So the lab takes a few days, so our test won't be probably closer to the end of next week. All right. So and tomorrow I'll give you the review sheet for chapter 9. I forgot. All right. So that's that. All right. Let's get this going here. All right, here's our notes.
How many entries do we have on Monday?
All right, so this last little bit of the chapter is about how we regulate our chemical reactions, and we do that by regulating enzymes. So it's titled Regulation of Enzyme Activity Helps Control Metabolism. Metabolism is the sum of all of our chemical reactions. So chemical chaos will happen if a cell's metabolic pathways are not regulated. And we regulate this a lot of times by switching on or off genes that encode for specific enzymes. So let's just do a quick little review. What does this mean? So this here. Genes. Genes are, where do we find our genes? And they're part of what molecule? DNA. So our genes are part of DNA. DNA, remember, is double-stranded. It has the sugar, phosphate, and the bases sticking out. And remember, the bases are A, T, C, and G, and the A's and T's hydrogen bond together, C's and G's, and so on and so forth. And so I'm not going to go right through that, but I just want to give a quick little review about DNA. And then it's the order of those bases, the A's, T's, C's, and G's, that determine what a gene codes for. So a gene is a segment of your DNA. So a small part of your DNA is a gene. Genes code for making what? Proteins. So if you remember that DNA is stuck in the nucleus, right? So, and your polypeptide chain is made where? What's my little circle here? This is the ribosome. So your polypeptide chain is made in the ribosome. And if you recall that DNA, the instructions in the gene is copied on a molecule of mRNA. The mRNA leaves the nucleus and goes to the ribosome, where the mRNA is quote-unquote read, and it's the order of those A's, T's, C's, and G's, and eventually we get our polypeptide, which is a chain of amino acids. Polypeptide is made here. So here's your polypeptide. Chain of amino acids, does this have a three-dimensional shape right now? No. So remember that then the polypeptide gets a three-dimensional shape by doing the alpha helix and the beta cleavage sheets and so on, and gets this three-dimensional shape, the interactions of the R groups holding them together. So let's say that this is your finished protein. This is an enzyme. An enzyme is a type of protein. So that's your enzyme. And so what this is saying here is the cell switches on or off genes. So you can see that if this gene is off, this process never occurs, and an enzyme is not going to be made in the cell. And so then it can turn it on, and then the enzyme can be made, and whatever reaction that enzyme catalyzes can happen. So let me give you an example of this. We have all of our DNA to make every single protein in every single cell of our body. So a cell in your skin, a cell in your stomach, a cell on your appendix, and so on. I have all the DNA to code for all of the proteins. But not every protein needs to be made in every cell. And even if it's made in the cell, it doesn't need to be necessarily made 100% of the time. An example, every single cell has a gene to make pepsin. Pepsin is the enzyme that digests protein in our stomach. So therefore, even our skin cells have the gene to make pepsin. But guess what? Our skin cells have a different job than our stomach cells. So even though our skin cells have the gene to make pepsin, do you think pepsin is made? No. And so those genes are turned off in those cells. In your stomach cells, you need to make pepsin, but do you need to always make pepsin 100% of the time? No. So therefore, your stomach cells have a way to turn on the gene to make pepsin. It makes it sometimes, and then it turns off when it doesn't need to be made, because that conserves energy, and so therefore you're not being wasteful. And so that's what that means, the cells switching on and off genes. All right, now sometimes by regulating metabolism or chemical reactions, we can control and regulate the enzymes themselves. So the next is about allosteric regulation of enzymes. This is the term used to describe cases where a protein's function at one site is affected by binding of what we call a regulatory molecule at another site. Allosteric regulation may either inhibit or stimulate an enzyme's activity. So when we look at allosteric regulation, it can be an activator or an inhibitor. Most allosteric regulated enzymes are made from polypeptide subunits. Each enzyme has active and inactive forms. The binding of an activator stabilizes the active form of the enzyme, and the binding of an inhibitor stabilizes the inactive form. So, activator activates the enzyme. So this enzyme catalyzes a reaction, so if it activates the enzyme, is the rate of reaction going to increase or decrease? It activates the enzyme. Increase. So this right here is going to increase the rate of reaction rate, and an inhibitor is going to make the enzyme so it doesn't work, and so this is going to decrease the reaction rate. And so what happens is this enzyme can be in these two different forms. So here we have this picture. Let me explain what we're looking at here. So here is your enzyme. And so your enzyme, notice it says there is an allosteric enzyme with 4 subunits. So each one of these right here are different subunits. So subunit 1, 2, 3, 4. What it means is this is made from one polypeptide chain, this is another, and this is another. So this is an enzyme that has the quaternary structure made from 4 different polypeptide chains. It's an allosteric enzyme. Why? Because it has sites different than the active site. So notice here that each of these subunits has an active site. So there's the active sites. And then it says here regulatory site, and it's this one of 4. And so each of the regulatory sites are between the subunits. So there's 4 of them. Another way to say regulatory site, I want you to write down is allosteric site. That's the same way, same thing. Allosteric site and regulatory site are the same. And so the enzyme here's in the active form. Why is it in the active form? Because the active site is in the right shape for the substrate to bind. This enzyme can catalyze how many reactions at one time?
4.
4, right? Because 4 different substrates can bind at one time. And that's in the active site. Now notice what oscillates between the active and inactive form. Inactive form, notice the shape change. And so the active site is changed shape, so therefore it's inactive because the substrate can't bind, and therefore no reactions occur. Now what makes them oscillate back and forth into the active and inactive form? And what makes them do that is the presence of the activators or the inhibitors. So let's look at this. If this is another molecule, an activator, notice that this picture shows the activator binding to the allosteric site. What that does is stabilizes and makes it so that enzyme stays in the active form. So no matter what, even if an inhibitor comes along, the inhibitor can't bind to another one of these sites and cause it to go into the inactive form. So it's stabilized in the active form, and this enzyme is now kind of what we call turned on. It can catalyze the 4 reactions at once, and it's active. Now what happens if there's an inhibitor that gets to that site first, the inhibitor changes the shape of this protein and changes the active site and stabilizes it in the inactive form so it doesn't work. And so allosteric enzymes can be in one form or the other. They're either active or not active. And we're going to see that, and I'm going to give you an example of this, is that often the allosteric enzymes are either activated or inhibited by parts of the reaction in which they catalyze. Question?
So the use for like allosteric enzymes and like inhibitors and activators are to regulate what is made and what is not.
That's right.
Okay.
That's exactly right. And so this is a process that our cells use that allows specifically to make reactions go or not. So I'm going to give you an example of that, and I'm going to write some things down on the overhead. I don't think you have enough room on your page, so I'm going to, if you'd like, you can get another sheet of paper out. But I want to give you an example of how and why an enzyme would oscillate back and forth and how it does that. And I'm going to use cellular respiration as an example. All right, let's review cellular respiration first. Let's make sure we're all in the right spot here. In terms of, we talked a lot about, I put cellular respiration on the board a lot this unit. Cellular respiration, an exer or endergonic reaction? This is an exergonic reaction. So we take glucose with oxygen and break it down into two molecules that have less energy in it, 6 waters and 6 CO2s. And since these molecules have less free energy in them, that means energy is released, meaning that this is an exergonic reaction. All right, so hopefully remember that from last week. So now I want to focus on this energy. So the energy released, we said some is released as heat, and that's why this is an exergonic reaction. It is increasing the entropy or disorder of the universe because some of that energy was once in the molecules of glucose there, now it's released in this disorder. But some of that energy is going to be used to do what?
An endergonic reaction to occur?
Yeah, it's going to do different kinds of work for the cell. And so let's talk about what kind of work that is. And so the energy released here, glucose releases lots of energy, but like things that you need energy for in the cell, like active transport or to drive an endergonic reaction, what molecule do we use to get energy for those things? It's not really glucose. What molecule? ATP. So let's relate this to ATP. So ATP, when we use it for energy for the cell, ATP is broken down into ADP and an inorganic phosphate, which means it's not attached to anything. Is this process exer or endergonic? This is exergonic, which means that energy is released. This is the energy to do work for the cell. So what kinds of work for the cell does this do? So this is what provides energy for active transport. This is what provides mechanical energy or energy for movement. We talked about like moving the transport vesicles and so on. And then also endergonic reactions. Endergonic reactions are reactions that need energy. And so ATP, since it's an exergonic reaction, can get that energy that's released can drive these endergonic reactions that need energy. That's, remember, called energy coupling. Do you remember that? All right. And so that's energy coupling. So what happens is that if our cells, if we keep breaking down ATP, we're going to have energy to do work for the cell, but we have a finite amount of ATP, so we'll run out of it. So therefore, we have to do the second part of this cycle, which is to regenerate ATP. This is a what kind of reaction? Ender or exer?
Ender.
This is endergonic. It needs energy. So where does this energy come from? Mainly from cellular respiration. So I'm gonna put CR for cellular respiration. So when we said over here that when we break down glucose and it releases energy for work for the cell, the energy, the work is making ATP. So that's what it actually does. So if you need ATP, you want cellular respiration to increase, right? Because then you'll have energy to make ATP. If you don't need ATP, let's say your cell has enough, we would want to slow down cellular respiration because we have enough ATP, and so on. And so that's what we're going to compare and look at today. So this is the background here. So let's talk about allosteric regulation in this process. All right, I'm going to attempt to draw an enzyme. All right, so I'm going to draw the allosteric enzyme kind of like in our notes here with the 4 subunits with the active site. So this we'll say is an active, this is the active allosteric enzyme. So it's active, it has the active sites, which the active sites are right here. So these are the active sites, all right, are the right shape. So let's write a few things. All right, let's pretend that this enzyme, let's say this enzyme helps break down glucose. So this enzyme helps what process? What's the breakdown of glucose called? Cellular respiration. All right, the process of cellular respiration. All right, so this is an enzyme that's going to help with cellular respiration. So let's give a scenario of the cell. Let's say if ATP production lags behind its use, I'm going to put blank accumulates in the cell. Take a minute and read what that says and think about ATP and think about what you would put in that blank. ATP production lags behind its use. If this is the case, if it lags behind its use, we have 2 reactions, breaking down ATP into ADP and a phosphate, and then we have putting it back together. Which one's occurring more, according to this? If ATP production lags behind its use, which one's occurring more, Shane? All right, ATP to ADP plus a phosphate, all right? That's right. So the production, this is the production right here, ADP and a phosphate. That lags behind its use, so this is occurring more and this is occurring less. Does that make sense? All right, so what would we have an accumulation of in the cell? We'd start to get a lot of, well, just looking at these reactions before we even go there.
So we have more.
Do you see where we're going to start to get an accumulation of ADP? Because this, so you have ATP in the cell, all of it's being broken down to ADP, so you now have all this ADP, but not as much ADP as putting it back into ATP, so you're going to start to accumulate ADP. Does that make sense? Okay, and so we're going to put here ADP accumulates. Now, my question to you is, what do we need to do in the cell now? So ADP starts to accumulate. So we're using up ATP. We're not making it as fast. Will we eventually run out of ATP if that's the case? Probably. So what do we want to do to cellular respiration, the rate? Increase it, right? And so what we do is so when this is happening, we need to make more ATP. All right, which means then we need to increase cellular respiration rate. The rate of I'm just going to put CR for cellular respiration. So we need this enzyme to work. We want the enzyme in its active state. So we want it to be in the active state. So guess what? ADP, in this case, as it starts to accumulate, the cell's getting more and more ADP. So what's happening is that ADP stabilizes this enzyme in the active site or acts as an activator. So ADP comes over here. Let's say that this is a molecule here. So this is ADP. It binds to the allosteric site, acts as an activator, which then increases cellular respiration in the cell. Which, if increases cellular respiration, is going to generate energy to make more. All right, yeah, so ATP levels go up in the cell, which is what we want to do. You guys follow me there?
Right, was that feedback activation? Or
They call that we're going to talk about it's positive they don't have it, it's not feedback. I like how you're making connections there. It's activating it. That we're going to learn a little bit later, there's positive and negative feedback, and so it's positive. Yeah. Okay. So it activates that, and then the rate of cellular respiration increases. So let's write here. So ADP acts as an activator. Which is what we just wrote here, which increases the cellular respiration rate and ATP levels go up. So now what's going on in the cell? You may have now a bunch of ATP, so your cell is making a bunch of ATP. Do you think it can become a point where, as it's making ATP and ATP starts to accumulate, that there's more ATP than you need? Yeah, that could happen too. And so as the cell starts to make ATP and it's being used, if it's being made more than it's being used, ATP can start to accumulate. And so let's write that down in that scenario. So if ATP is made more but stops being used, or its use slows down. ATP can accumulate in the cell. All right, so now if you have ATP accumulating in the cell, do we need cellular respiration to really keep going?
No.
No. So therefore we want to slow cellular respiration down. So what do you think ATP acts as?
The activator.
Not the activator. The inhibitor, because we want it to slow down, so we want to inhibit that enzyme, and so it acts as an inhibitor. So now in this case, ATP, we need to stop making ATP. So what does that mean? We need to slow cellular respiration down. And how we do that is ATP acts as an inhibitor. So if we draw a picture here, so this is your normal enzyme, right, with the normal shape of the active sites. So this is normal, this is the active enzyme, right, Max? This is the normal active enzyme. And so what happens is if you start getting ATP accumulating in the cell, so I'm going to draw ATP as these little diamond-shaped molecules. So ATP is accumulating in the cell. So what's going to happen is this ATP, as it accumulates, can bind to that allosteric site. When they bind to the allosteric site, what does it do to the enzyme? How does it act? This act as an inhibitor? It's going to change the shape. So I'm going to do some crazy shape, all right, just to illustrate here. So my shape change here of my enzyme. Here we have our ATP bound here. So ATP binds to the allosteric site. ATP is an inhibitor. The inhibitor, the shape changes. Enzyme changes shape, which means that cellular respiration slows down. Which means that what happens to the production of ATP? ATP production decreases. And then we could go round and around. This can happen again and again. So now, so ATP we had, just to give you a picture here. So there's a whole bunch of ATP in the cells. So you have a whole bunch of ATP in the cell. And you still might have a few ADPs. It doesn't mean that there's no ADPs in the cell. So you have all this ATPs. This ATP binds to the enzyme, inactivates and stabilizes it in the inactive form. So we're not making any more ATP. But what happens to the cell is the cell's now going to start using some of that ATP. So now if the cell starts using this ATP, it'll break down into ADP and a phosphate. ADP and a phosphate as it's using it, ADP and a phosphate, and so on. And it starts breaking it down. Then what's going to happen is your ADP concentration is going to go back up. And when it does this, this is reversible. So it actually, when the cell starts using up the ATP that it has so much of, it can actually take this off and use this ATP and break it down into ADP. When that happens, the enzyme goes back to the normal shape. And now, when that happens, you've got now all this ATP broken down into ADP because the cell's been using it for energy. This ADP, now we start the whole thing over. That ADP binds to the allosteric site and keeps that enzyme in the active site, or in the active form, I should say. And cellular respiration occurs. It begins making more ATP until ATP becomes in high concentration, and the whole thing goes back and forth and back and forth. All right? Does that make sense? Okay? Question? Yeah, I mean, it's not perfect. Like, in terms of, when we're talking about turning these guys on and off, there's never going to be 100% of, ATP and so on. So here there might be some ADP molecules and so on. And remember that these guys don't have eyes either. So these molecules are just randomly bouncing around the cell. And so what happens is, this is the reason why it's so important that if one, like in this case here, when ATP is so prevalent, if one ATP molecule gets into that site, it stabilizes it, so that now, even if an ADP molecule hits it, it can't activate it. You know what I mean? Because it is possible to have ADP molecules, even though you don't need to make more ATP, you can have an ADP molecule hit the enzyme and activate it, so that is possible, but it's not likely because the concentration of ATP is so much. But you're right that it's, you know, it's imperfect, but so we oscillate back and forth between there. All right, so that gives you kind of a real example of what I'm talking about with the same enzyme going into the active versus inactive form, and the reason why, that it's actually the molecules involved that are kind of guiding which direction these reactions are occurring. All right, so going on here, so adding on to allosteric regulation is this thing called cooperativity. It's a form of allosteric regulation that can amplify enzyme activity. So what this means is that if a substrate binds to one active site, it stabilizes and so what happens is the enzyme can't change shape once the substrate binds to it. And that's also helpful as well. So what happens is if a substrate binds to an active site, it keeps the enzyme in the active form, which means that other substrates can bind to it. So they're cooperating kind of together. One binds to it and says, hey, I've got it stabilized for the rest of us, and it keeps it stabilized, and that's called cooperativity. All right, and the last thing and we've already kind of talked about this, is something called feedback inhibition. Another way to say this in the title, I'm going to have you write or negative feedback. It's also what's called negative feedback. So literally after where it says feedback inhibition, I'm going to give you time to fill in the blanks. Write or negative feedback. So in feedback inhibition, the end product of a metabolic pathway shuts down the pathway. This prevents a cell from wasting chemical resources by synthesizing more product than is needed. So you have a picture here that shows this, and it's kind of like the shoe factory example from our homework. So let's look at the reaction here. You have threonine. There's an enzyme number 1, that catalyzes the reaction of threonine into whatever intermediate A is. Then enzyme 2 catalyzes it into B, 3 catalyzes it into C, enzyme 4 catalyzes it into D, and enzyme 5 eventually catalyzes it into the end product. So this is the pathway here. And so what happens is that sometimes our cells need isoleucine and sometimes not. And so sometimes, isoleucine is an amino acid, by the way. So it can be used to make polypeptide chains and so on. So if your cells need isoleucine, this will never build up because the cell will take that product and use it. If isoleucine is not needed, it starts to build up. And so what it does, if it starts to build up, is it actually acts as an inhibitor to that first enzyme. So notice here it binds to an allosteric site. Of this first enzyme, changes the shape so this enzyme doesn't work. And therefore, step 1 doesn't occur. If step 1 doesn't occur, then the rest of it doesn't occur. And so the buildup of isoleucine actually stops its own production by stopping that first enzyme. What happens next is the cell needs isoleucine, then it'll start to use it, and when it uses it, it takes it off of the enzyme and it's used up by the cell. The enzyme goes back to its normal shape, and now the pathway can occur, and that would make sense because it's now using isoleucine, so we need to make some more of it. Then as the cell ceases to use isoleucine, it builds up, it inhibits the enzyme, and so therefore this pathway is turned on and off depending upon if the cell is using the end product isoleucine. Does that make sense? All right? And so that is feedback inhibition. This is really simple.